from that digesting food. So get basically everything it can out of there, so that pretty much all you're getting rid of is the waste, the undigestible material, and not the stuff that is worthwhile to the body. And even water and electrolytes are worthwhile. Now they're not, you're not going to get much nutritional value. You're not going to absorb many small, organic molecules here, but you are going to do a lot of that other kind of stuff, reabsorbing water and electrolytes. And this is an important thing. And I'm sure most of you have had some sort of an intestinal infection, and one of the symptoms of intestinal distress, of course, is diarrhea. And what happens in diarrhea is sort of in part, is the colon kind of shuts down, and from the sense of absorbing stuff. Your colon says, there's something in my body that shouldn't be there, I just want to get it out of there. And it does that by just not absorbing water anymore. It dumps water into the colon, and that gives you the watery excrement, and that's the kind of idea behind it, is to get rid of all that kind of stuff. But obviously that's not the case in the normal situation. Now, when you look at the histology of the colon, you're going to see this is the mucosal layer up here. You're going to see all these folds of the very tightly joined together, or tightly opposed to one another, folds of the mucosa. The epithelium is fairly uniform. It's a simple columnar epithelium with lots and lots and lots of goblet cells. Right? The one thing the colon does want to do is to provide mucus. The mucus is going to be important for lubricating the waste so that it comes out a little bit more easily and flows through the intestine a little bit more easily. And you see, basically, it's all these little pits, all these little pit glands. Right? So, there's no sort of floor here. We're not going down into below the surface. It's all, this is it. The lamina propria is in between the little fingers, and then we have the muscularis mucosa down here, just beneath these epithelial folds. Right? So it's a fairly, histologically, it's relatively simple. There aren't any, sort of unique kinds of things in here. Here's a LOMAD picture of the colon, and you can see all these folds here. You can see the muscularis mucosa there. This would be the submucosa, and then the muscularis layer. The one thing that is particularly evident, depending on where you're looking in the colon, is that the muscularis externa becomes a lot more pronounced. Right? It's a much bigger, kind of a hardier muscularis externa, because it's there to squeeze the waste out. And that's a little bit of a more difficult job. Requires a little bit more strength to do that than just sort of squishing this gooey food waste through this food material through your intestine. It's a little bit more solid. Requires a little bit more effort to push it along. So you're going to see a thicker layer of musculature there. And in many places in the colon, you'll actually see 3 layers of musculature as opposed to the 2. In our histological sections, that's a little bit difficult to see, but you generally speaking will see some larger, some of these larger chunks of muscle underneath the submucosa to, kind of squeeze things and move things along. And of course, there's a lymphatic nodule. Right? We find those there in our intestine, in our large intestine as well. Other things that we find in our intestine that are not present histologically is lots of bacteria. There are many, many millions and millions of bacteria, actually billions of bacteria, in your colon. Right? Many of the bacteria that live in your body live in your intestine, specifically in your colon. They're actually very important to you. We are just sort of now learning a little bit more about the bacterial flora that's in our bodies and how that can impact our sort of normal operations. And in fact, it's been found that people with digestive disorders, things like Crohn's disease or ulcerative colitis, which largely affect the colon, it's being found that a lot of these can be at least partially caused by sort of a bad mix of bacteria in your colon. And one of the things that they can do is take a person who has these disorders and inoculate them with colon bacteria from a healthy person, And what happens then is, in many cases, their symptoms are eliminated or at least relieved by having a sort of a better complement of bacteria in their colon. It's a little bit kind of gross when you think about it, right? Because one of the easiest way to do it, to use the sort of proper terminology, is literally to do a fecal transplant. Right? So it's kind of gross the way it sounds when you think about it that way, but it's actually a really good idea, and it really makes a lot of sense. So, just something to think about. Close-up picture of the colon. These are the crypts. So these are these glands that go in, and you can see the epithelial cells here. Lots and lots of goblet cells sort of cranking out a whole lot of mucus there. Just to back up a second, our fecal material is actually largely bacterial cells. Right? A lot of it's food waste, but an equal portion at least is also bacterial cells. Now, at the junction of our large and small intestine, we have the cecum. The cecum is a blind pouch at that junction, and a little finger-like projection off of that cecum is this thing here. This is the appendix. Proper name is the variform appendix. And when you look at it, this is a cross-section through the appendix. You can see the muscularis layer submucosa. It's very, histologically, it looks very much like the digestive tube. It is a branch off of it. But what you see in this is a lot of lymphatic nodules. And so what happens with the appendix is that it's largely a segment of the intestine that has a very big immunological function. Excuse me. Obviously, it's not essential. We can live without it. As you know, in some people, this can get infected. If it does get infected, it can develop, swell up with fluid, and if it bursts, the fluid and the bacteria that are growing in there spread throughout your body cavity and can lead to sepsis, which is pretty dangerous. But generally speaking, you can just get rid of it before it bursts, and then you're okay. But this is kind of what it looks like. So it doesn't have a huge function in us, and we can certainly live quite well, and really, taking it out has really no impact on the individual. But it is there, and it probably does play some role in the immune system. It's just not something that's essential at this point. Other organisms, specifically ruminants, do have an appendix, and that appendix is functional. And part of the function of that appendix is it kind of serves as a pouch of reserve for the bacteria that help them to break down the cellulose in the grains that they eat. So that's what that's all about. That's our appendix. All right, so now we're looking at the opposite end. So we went from the early oral cavity, so the beginning of the food coming into your body, and now we're finishing up here with the other end of things, the anus, where the waste comes out. So we're looking here at the rectum, which is the last section of the colon. You can see some lymphatic nodules there. Not shown here, but if you were to look, and we have slides of this, if you look around to the periphery of the rectum, what you will see is apocrine glands. So I encourage you there's one slide today that will show you these guys. If you look off to the side a little bit, you'll see these apocrine glands. They're going to be just tubules, so they're just going to look like sort of circles of cells. They tend to be a little oblong or misshapen, but they're pretty big. Right? And these are the apocrine sweat glands, the ones that produce the oily kind of secretion. And they're going to be found in this area here. And what happens here is kind of, if we think about what happened with the epithelium as we went into the oral cavity, right? We have keratinized stratified squamous epithelium on our skin. You go into the oral cavity, and we start losing that keratinization. We go down through the esophagus, and it's still stratified squamous, non-keratinized. And then we get to the stomach, and we go to a simple columnar epithelium. That simple columnar epithelium is retained throughout the intestine. Well, down here, on our way back out, we got to undergo that transition again, right? We have to take this region here on the inside is going to be simple columnar, and then of course out here on our bottom we have skin. So we have keratinized stratified squamous epithelium. right? So this right here is going to be the transition zone. This is musculature here. The musculature here, of course, is the anal sphincter, so it keeps everything closed up and keeps the feces from just sort of dropping out whenever it would kind of want to, which is good, obviously, for hygiene. So, but this is what we see. And this is a picture of that area, right? So here's the anal sphincter muscles here, and here is the internal zone. So this would be up here, the colorectal zone. That's looking more or less like the colon. The ATZ is the anal transition zone, and then the SQZ is the squamous zone, and then so we're transitioning from one to another, simple columnar to a stratified cuboidal, and then eventually to a stratified squamous epithelium. So right here, this portion right here, this would be the actual anus itself. That would be the edge of it right there. So kind of looking at things coming out the other end. But it's all contiguous, right? So we got to kind of flow from one to another. And just as we saw transitions between the esophagus, the stomach, the intestine, and so forth, we see changes particularly in the in the epithelium as we go through here as well. So that's kind of what we're looking at. All right, so we'll switch gears here now on that one and move our way on to looking at the accessory glands that are going to play an important role in the digestive process. And we know that the digestive tube does a lot of the work of digesting and absorbing our food, but it doesn't do all the work of digestion. We have other organs that are going to help us out here, other organs that are going to help with the digestive process, as well as a number of other things. And we can see a couple of them here. The one we're spending a little bit more time talking about today is the liver, the big red thing there. And we'll also talk about the gallbladder, the little green pouch right there. That's relatively simple. And we'll also talk about the pancreas as well. Right? 3 accessory glands that are going to play various roles in assisting with the digestive process. Now the liver is actually kind of an amazing organ. It is our largest internal organ. If you remember back to, if you took Bio 110 here, if you remember back to dissecting the rats, the question I always get when doing that from students is they'll open up the abdominal cavity and they'll see this big sort of dark red thing right here underneath the rib cage. Like, what's that? That thing's huge. And nobody knows what it is. And then eventually I tell them it's a liver, and they go, oh, okay. Because they don't realize how big the liver actually is. The liver is a rather unique organ for a couple of reasons. One, in terms of its breadth of functions. It does a whole host of different things. The other is that it is probably, I think, the only internal organ that has the capability to regenerate. You may have heard of people doing live liver transplants or live liver donation. All right? The liver is one of the few organs. Now, you can't cut it all out and then have it, sort of grow from nothing. You can donate a portion of your liver to a recipient. You can't donate all of it because if you didn't have liver function even for a little while, you'd die. But you can donate like half your liver or so, and then the other person can have the other half, and your liver will grow back for the most part. Right? It's pretty much asymptomatic. You have to deal with some issues initially, but after a little while, the liver kind of grows back and you're in good shape again. Right? So kind of a unique situation. Now, the liver, like I said, does all sorts of stuff. There's this whole list. If you look at the list in the handout for this class, you can see tons and tons of different things that the liver does. It makes various proteins that are involved. That are important for our body, like albumin, a major protein in the plasma of our blood, is important for osmotic balance and buffering the pH of our blood. It makes clotting factors, proteins involved in the clotting process, lipoproteins, glycoproteins for iron transport, globulins that are found in our blood as well to help maintain osmotic balance. It's involved in vitamin production, A lot of iron stuff takes place, iron metabolism takes place in the liver, a lot of iron uptake using proteins like ferritin take place in the liver. A major function of the liver is detoxification. So when our body gets nasty substances in it, whether we take them, which happens sometimes, or whether even just normal metabolism often produces toxic substances. And the liver's there to modify them, to detoxify them, and then get rid of the waste. So the liver's going to play a really big role in that sort of process. So it's another organ that's going to, do a lot of monitoring of the blood. We saw how the spleen works to monitor the blood to get rid of old red blood cells and platelets. The liver is going to basically sort of monitor the serum and sort of take all the junk that's in the serum that shouldn't be there and get rid of that. So when we look at the liver cells, we'll see that they're going to have lots and lots of ER, especially smooth ER, because the smooth ER is involved in detoxification. So that's kind of an interesting thing. So some of you may have heard this little anecdote before if you had me in Bio 110. But one of the things I always tell the Bio 110 students when I'm giving them the overview of smooth ER function is that, I point out how the liver is involved in detoxification of substances. And one of the things that happens with this is that if you take a look at the liver of a person who takes a lot of drugs, whether it's drinking a lot of alcohol or injecting heroin, it doesn't matter. Whatever kind of nasty substances you put into your body, they're going to be detoxified by the smooth ER in the liver. Well, one of the things you may be familiar with is a concept known as tolerance. As you take more and more of a certain drug, you develop tolerance. So in other words, when you take the drug for the first time, you take a certain amount, you get a certain feeling from it, right? But after a while, you develop a tolerance to it, so to get that same initial feeling that you got, you're going to have to take a lot more of the drug. And what's happening there in part is that the smooth ER in your liver cells, your hepatocytes, is growing. So these liver cells, which are normally pretty full of smooth ER, are making even more smooth ER, so they're getting better and better at getting rid of this stuff. So it's kind of like it's like the liver's getting in shape or something. It says, oops, I keep getting more and more of this stuff. I got to do a better job of getting rid of it. So it makes more and more smooth ER. So you need to your body gets better at degrading the stuff. So you need to take more of the drug to get the same feeling. That's where the tolerance comes from, in part. Right? There are other factors, but that's one of the things that it leads to. Metabolically, there's all sorts of stuff that happens in the liver. Glycogen production for energy storage, lipid metabolism, cholesterol metabolism, urea production, amino acid synthesis. A lot of these things happen in the liver. So all sorts of good stuff going on here. It's one of the things I always, when I was younger, I know on occasion my mom would cook liver, which I always hated. But now as I know more about what the liver does, I keep thinking, why would anybody want to eat this? Right? This is the garbage dump of your body, right? This is where you are taking all of the junk, all the nasty stuff, and you're sending it here to get rid of it. Why would I want to eat that? It's full of cholesterol, right? Well, yeah, it is. But you have to, if you think back to, a kind of a different era in the history of humans, well, yeah, I could look at it now and say, oh, there's no way I'm going to eat that. But, if there's nothing else to eat, you're going to eat it, right? And if you're not like us, where we have an abundance of food, typically, that cholesterol, that doesn't matter. That toxic stuff, whatever. It's that balance, right? The nutrition versus the negative from the toxic substances. There's no contest there. The nutrition's going to win. Plus, it's a really, really good source of iron, as you might imagine. So if you're malnourished, well, sure, you're going to eat the liver because it's, there's a lot of good stuff in there that you need. But if you don't really need it, well, not an organ I would choose to eat if I have the option. The liver is also involved in bile production. So one of the things that happens as a result, or there are a couple of things, but as a result of all of this detox, what happens during the detoxification process is that the liver breaks these things down, breaks them down into non-toxic products, and then puts them into the bile. The bile has other stuff in it, but the bile is going to play a role in the digestive process. The bile is going to be secreted, it's going to be sent to the gallbladder, and then eventually to the small intestine. And the bile is going to be put into the small intestine, and the products of the bile salts and other things, are going to aid in the digestive process. In particular, they're going to be really helpful in digesting fats. So there's emulsifiers in bile that's going to take the fat and break it down into tiny little fat droplets that are going to be more readily absorbed by the system. All right. So there's all that kind of stuff going on there. All sorts of other things are going on in the liver. It has some endocrine functions. It makes vitamin D, pyroxin, for example. All sorts of stuff all sorts of things are made by the liver. All right, so let's take a little bit of a closer look at how the liver is put together. The liver is separated into these lobes, and this is sort of a traditional way of looking at the liver. What we have is the sort of reddish part here is the main part of the liver that contains mainly the main cells of the liver, the hepatocytes, which are the cells that are going to be doing most of the work of the liver. And we're looking here at the blood supply to the liver. We have an artery and a vein. The liver is actually, circulation-wise, this vein that comes into the liver. Now this is not the main one, but this is a branch. These are branches from the hepatic vein that comes into the liver. The hepatic vein actually passes by the intestines and then goes right to the liver. So the vein picks up all the digested food molecules from the intestines and then heads to the liver. And the point of this is twofold. One is you kind of want the liver to have the first shot at this. One is if there's anything nasty in the food, it can go to the liver right away and get detoxified. Two is your liver does a lot of important stuff. So you kind of want to give it a pretty good shot at those nutrients. So that's part of that process. So what's going to happen is these veins are going to feed in, and then the arteries are going to feed in as well to the liver, and those are coming from the other half of the circulation. And they're going to come in and they're going to mix together. And the liver, the blood will mix and flow through a hepatic sinusoid, which is some ways similar, at least conceptually, to a splenic sinusoid. And it'll interact with these hepatocytes and get they'll do their thing in terms of filtering the blood. It'll flow into this central vein here that's in the center of the lobe, and then go down and out. All right, so it kind of comes in here, it percolates through the lobe of the liver, and then goes back down. And you'll see also associated with this is a bile duct. We'll look at how the bile duct comes into play in a little bit. But the bile duct actually works in the opposite direction. So these, what we'll see that are called hepatic cords, are going to filter stuff out of the blood, and then they're going to make bile. And the bile is going to go through these little canals out to the bile duct. And you can see the arrow going down. It goes down and out, and ultimately to the gallbladder. And so here you can see more of a close-up picture of how this works. So here we have the arteries coming in, the veins coming in, and these are the hepatic cords here. And these sort of lighter red-colored spaces here are the sinusoids. Right? So these blood vessels come in. You can see a couple coming in here. Here's the sinusoid. Right? The blood goes through here into the central vein and then on out. You can see all these holes are where these other connections would be from the chunks of the liver that have been cut away would be. All right. Now this little line in the middle, we have a double row of hepatocytes, but the little line in between is actually what's called a bile canaliculi, or bile canaliculi, for the plural. All right. So it's a little tube that leads out from the hepatic cord to the bile duct. So it's going to lead out this way, and then the bile will come on out. All right. So this is the kind of thing, histologically, this is about the level, at least diagrammatically, that you're going to be looking at it. Right. You'll be able to see the central vein. You'll be able to see the hepatic cords and the sinusoids, and you'll be able to see these the portal vein, hepatic artery, and the bile duct, which are located in clusters called the portal triad at various locations around the liver. So you'll see the lobe, it's arranged in this nice little, what's it, 1, 2, 3? It's an octagon. And basically, roughly speaking, it's not entirely true, but roughly at most of the corners of this octagon, you're going to find these portal triads. It's not quite that regular, but it's something approximating that. All right, so the blood flows in through here, percolates through the hepatic sinusoids, goes into the central vein, and comes on its way out. Now here we can see a couple of pictures here. This is a picture of the pig liver. The nice part about pig liver is that it has connective tissue surrounding each of the liver lobes. All right, and that's what you see here. This is a picture of a human liver. There's a dashed line drawn in, but there is no connective tissue there. So the pig liver is nicely separated into individual lobes. The human liver and most other livers are not. So they kind of have this sort of uniform appearance. But this is the central vein here, kind of in the middle, and these are all the portal systems here. And if you were to look closely at one of these, you would see at least 3 ducts. You'd see artery, vein, and bile duct, amongst other things. Sometimes you'll see a lymphatic vessel as well. So this is the kind of thing that you would see here. So this is more like what you're going to be seeing with the liver. So here, all this red stuff, those are the hepatic cords, and the white areas in between are the sinuses. Right? That's what we're going to be looking at. So here's another sort of general picture of a liver. This is more along the lines of what you're going to be seeing. So here's the central vein, the hepatic cords and sinusoids, and you can see the portal system here. And those are some of the portal systems. This looks like a larger bile duct. The bile ducts are this is another one here. The bile ducts have a cuboidal epithelium, so they're going to look a little different than arteries and veins because of that cuboidal epithelium. So here's a close-up picture, and you can see clearly the hepatocytes. Here's the central vein, and you can see, like right here, here's one of those sinusoids opening up into the central vein, and you can see the cords of hepatocytes with the sinusoids in between. And then over here, we have the portal system, at least one of them in this picture. And so this is the portal system. This little guy right there, that would be the bile duct. It's small, and it's smaller. These guys are going to be a combination of arteries, veins, and lymphatic vessels. That's the kind of thing you're going to see here. Low-mag picture, once again showing you the central vein and all the other thing. This one is fairly well separated into individual lobes, not necessarily typical of a human liver. This particular diagram or micrograph is stained for reticular fibers, and that's what we see here. These dark lines are the reticular fibers, so that you can see the surface of the cell that is going to line the sinusoid has these reticular fibers associated with them, and that's kind of what we see here. It's kind of a nice picture outlining all of the different sinusoidal spaces, and of course the central vein right here. This is what's commonly referred to as the portal triad. That's what we see here. So this is the bile duct. This is obviously a high-mag picture. This is the bile duct, so you can see the cuboidal epithelium here. This is the vein, and that's the artery. And you also frequently have a lymphatic vessel associated with it as well. So that's just one of the things we're going to be looking for with with regard to the liver. So here's a sort of diagrammatic representation of the blood flow through through the system. Here we have the hepatic artery and the artery and the vein coming in. Right, they all join together, and the blue here would be the sinusoids draining into the central vein. And then over here we have the hepatocytes. That's the double layer of hepatocytes, and you can see the little green arrows show you what happens with the bile as the blood flows through. The liver cells, the hepatocytes, take in whatever they're going to take in, do their thing with them, and then they put the waste product and other products into the bile. And the bile flows outward away from the central vein and into the bile duct that you see here. So this is a close-up picture of that area. So this is a hepatocyte here. This is a red blood cell. But right in here, this is the sinusoid. Right? The EN refers to the endothelial, the endothelium. So this is the endothelium of the sinusoidal blood vessel. And this endothelium is broken. You can see a little gap here. There are gaps all over the place in the endothelium. It's very, very porous. Now, obviously not porous enough to let a red blood cell through, but porous enough to let all the molecules that are dissolved in the plasma to get through. So this is just the very edge of the hepatocyte here. And these hepatocytes have all these finger-like projections sticking off of them. And that's what we see here. These are all those finger-like projections. And what it creates is this little zone at the surface of the hepatocyte where, first of all, we have all sorts of surface area, because all those finger-like projections sticking off of the cell give it a huge surface area. And then we provide this space for that fluid to circulate through. So as the blood flows through, the liquid portion of the blood squeezes through the openings into this perisinusoidal space over here, depicted by D, which is called the, where is it here? It's on this, yeah, space of Dissy. That's this here. So it gets in there, and that's where the liver cells can see what's in the blood, take in what they need or what they want, and do their thing, whatever it may be, absorbing nutrients, getting rid of toxic stuff, releasing things into the blood, whatever the case may be, it's going to be happening all along here. Okay, so that's how that's going to work. What's the purpose of this one? So here's a close-up picture or a diagrammatic representation of this, right? So here is the blood circulating through. These are the endothelial cells of the sinusoid. This is the space of Dissy, and then these are the hepatocytes with all their finger-like projections. Over on this other side here, we have the bile duct. Right? So the cells are going to do their thing and then get rid of the products over. Actually, this is the bile canaliculus right here. This is just the other side of that. But these are the joints between them that are going to kind of come together. Right? This is where the bile is going to run through. So they'll put all the bile, all the waste products, all the bile components into here, and then they'll go through in that direction. Right. Electron micrograph of a hepatocyte, and what you see, there's the nucleus. Just look at the cytoplasm of the cell. Right? And you're probably not very used to looking at electron micrographs of cells. The dark things here are all various different sections through through mitochondria. But what you see in the cytoplasm is all these little circular things. That's all membranous material, tons and tons of membrane, right? And this tells us that really it's full of ER, right? All these little circles are basically smooth ER, right? So we have lots and lots of smooth ER in these cells, and that's what this picture is showing you. These little dark spots are glycogen inclusions. So our liver cells like to store energy in the form of glycogen, and this is where that takes place. Here's a biocanaliculus, right? This little tube right here, you can see that. That's running in the middle of the hepatocytes, and it's going to head its way out to the bile duct. All right. So from the liver, what's going to happen is it's going to feed into the common hepatic duct, and eventually spill its contents into the gallbladder. And the gallbladder, largely what's going to happen is the bile that's produced by the liver is going to be concentrated. So the bile duct here is located on the underside of the liver. It holds about 50 milliliters of fluid, and it's this kind of hair-shaped structure. Very simple structure, right? We have a very thick kind of muscularis layer, which is involved in squeezing the bile out, and the adventitia on the outside, and then the mucosal layer on the surface. So histologically, it's not terribly complex. You'll see a bunch of blood vessels in the gallbladder, and here's a closer up picture, showing the epithelium, and then the musculature underneath, as well as some connective tissue. Right? So nice columnar epithelium here in the gallbladder. So not a whole lot to see in the gallbladder. There are some glands that are going to produce some of the components of the bile that is produced by the gallbladder. Yeah. So largely it's just holding stuff and concentrating it for later use. Right? So this is what we see with the gallbladder. Now, so not too much to say there. Here we have the pancreas, and the pancreas is another pretty interesting organ. It has a couple of different general functions. The function that we have, the main function of the pancreas, at least volume-wise, is in producing digestive enzymes. Digestive enzymes are made in the pancreas, ultimately secreted into the duodenum through this duct, which also brings the bile to the duct. Right? So the pancreas is going to be making a lot of these digestive enzymes. Right? They're coming from the stomach. The stomach's right up here. Right? This is the duodenum, so the stomach comes in here, and here's our duodenal arch. The duodenum is where the pH of the digesting food is going to be converted from the acidic stomach to the slightly basic intestine. And as you might imagine, the enzymes that function quite well in the low pH of the stomach are not going to function at all in the much higher pH of the duodenum. Well, these enzymes are made in the pancreas and secreted into the intestine here. Right? So the stuff comes in, the pH is raised, and then we add enzymes to help absorb that kind of stuff. Now, when we look at the pancreas, you'll see something like this. This is a nice little picture. This is a pancreatic acinus. Right? We have these purple cells with our nuclei, and there's a little duct. So these cells are going to be making the digestive enzymes, dumping them into this intercalated duct, and on out. Now this may look kind of similar to another organ that we looked at before. In many ways, at least what's called the exocrine portion of the pancreas that makes digestive enzymes looks very, very similar to the parotid gland. Right? It looks very similar to the parotid gland. So you can see these little purple clusters of cells connected to ducts. Right? It's going to be different. You'll see some differences though as well. These cells make enzymes. So all these little red dots here, those are called zymogen granules. Those are compartments containing the inactive digestive enzymes. The enzymes are made initially in an inactive form, so they don't digest each other or the cell. And then once they're secreted into the duodenum, they'll be activated. All right, so they're secreted, they go down, collect into these bigger ducts, and eventually end up in the duodenum. And this is kind of what it looks like. These are the pancreatic acini, right? These purple clusters, and then you'll see some ducts as you scan around. So this is the kind of thing to look for, these purple kinds of ducts. So that is the exocrine portion of the pancreas. But as you probably know, the pancreas also has an endocrine function. It makes hormones. Most notably, the most well-known hormone that is made by the pancreas is insulin, involved in regulating glucose uptake. And if you look at this low mag picture of the pancreas, you can see a nice artery here. But you see all these little kind of pale spots. Right? The darker purple areas, those are the areas that are involved in making digestive enzymes. The lighter areas here, those are the islets of Langerhans. And those are the endocrine portions of the pancreas. They're the ones that are going to be making hormones. Three hormones: insulin, glucagon, and somatostatin. And you can see a close-up picture of these here. So here's our pancreatic islet here, the nice lightly stained area here, routine H&E section. And then this is a special stain that highlights the cells producing glucagon. Those are these darker cells over here. Right? So basically, three hormones are produced by the pancreas. The one that you're most familiar with is insulin. Insulin is, of course, involved in increasing glucose uptake by the cells. And the vast majority of the islet are the pancreatic beta cells that make insulin. The A cells or alpha cells are the ones that are stained here. Those guys are going to make the hormone glucagon, which, if you recall, is the opposite of insulin. Right? Insulin says take up glucose. Glucagon says get rid of glucose, release glucose. And then there's another hormone that's made by a small number of cells within the islet called D cells that's going to make a hormone called somatostatin, which is going to regulate the release of human growth hormone. Right? So this is that's how the islet is put together. Here's another picture. You can see the exocrine portion here in purple, and here's our nice light endocrine function in our islets of Langerhans. It's a different color, and it also has a different sort of texture to it that you can see in this picture. And this is just showing you a sort of diagrammatic representation of it. The A cells are around the outside, these are the beta cells, and there are just a couple of the D cells in there making the somatostatin. That's kind of what we have. All right, that's our kind of whirlwind tour of the